Good morning, AP Bio. Hope you had a good weekend. For today, a few updates and then a little bit of additional information on mutations that should help you with your protein synthesis simulation packet, the questions at the end. All right, so first up, a little bit of the, just basic info I wanted to share with you. I am in a post grade, so make sure you're looking at those grades and I'm getting everything in. Some of you have emailed a few things just like last night that I haven't quite gotten in yet, but look and make sure things are looking updated. Okay. Then for the next couple days, I'm going to be listening to your protein synthesis simulation. And we're going to do that through Google Hangouts and with the video so I can see it. Now, all you need to do for that is be online and ready, and I will invite you at the appropriate time. Now, if one runs a little over something, it might be a little late, but just be ready to answer, and then we'll start from there. Okay, have your computer set up and all the things out so that I can kind of see that as we talk through. And again, the important bit is talking through protein synthesis so we make sure we understand all the steps and being able to visualize it with your cutouts. All right. So I will see you all and talk to you all soon through that. Now, while we're working on that, another thing you can be doing is actually finishing the packet of questions that it comes with. Okay. And as you flip through past the information on how to make the simulation, which is what we've done so far, there's like a series of questions. And it's kind of like typed out crazy, so it takes up so many papers. One thing that I want to mention is a lot of it is on mutations. And so when it goes through a mutation, it asks you to transcribe and then translate the mutant strand. So it asks you to go from DNA to RNA and then RNA to amino acids. Well, just so it's not too tedious, you do not need to transcribe and translate everything. You can just do where the mutation occurs because they give you the normal strand. If it's a mutation of just one base, you can just transcribe that codon, that 3-base sequence, and just translate that one different amino acid. Now, if it's a mutation that changes every amino acid after it, you need to translate all the mutations after that, or all the changes in the amino acid sequence. Okay, and so with that, I had a little additional information. You could add this at the end of your notes, just on the back page or wherever, but find a little spot and it'll always be here for you to access as well to look at this additional information, which is basically on mutations. So here we go. A mutation is simply a change in the DNA sequence. It can be bad, it can be good, it can have no effect at all. It is just a random change in some part of that DNA sequence. Okay, so what are some ways that DNA can change? Well, one base can be different. All of a sudden, like in the sequence I have below, a C is a T. All right, that is called a point mutation, when one base changes. Okay, so in our 3 billion bases, one base changed. And if it is in a bit of DNA that codes for a gene, okay, it can potentially affect that protein. So it is a point mutation when one base changes. Sometimes we can insert or delete a base. So an insertion mutation or a deletion mutation, and it is exactly what it sounds like. And it can be one or more bases. Now, if it adds or deletes just one base, you can see how everything would shift over after that point, meaning the reading frame would change. So every codon after it would be different. When an insertion or deletion causes all the codons after it, and therefore all the amino acids after it to change, we call it a frame shift mutation. Okay, and the last one is called an inversion. It's where one chunk of your DNA gets flipped. Lots of times this happens when the DNA is coiled so tightly, if it pinches off and breaks and like reattaches differently when it uncoils, it can cause an inversion. Okay, and so what this causes in the amino acid sequence is a mutation as well. So something can be a point mutation, which is also a silent mutation. Okay, so a silent mutation is when there's a change in the DNA sequence, but it doesn't cause a change in the amino acid sequence. If you remember back to the wobble and how lots of times the third base in a codon doesn't change what amino acid it codes for, that could be something called a silent mutation. Okay, and so it's going to have no effect. So it'll be passed on in the genes, but it's going to have no effect in the amino acid sequence in the proteins, so it kind of doesn't matter. All right, a mutation can cause a missense mutation. And this is where the change in the DNA changes the amino acid sequence. Maybe it just changes one amino acid, or maybe it changes a huge portion, like a frame shift would change all the amino acids after that point. So this is going to cause a change in the protein, and depending on how different the amino acids are from each other and how many are different, will cause more or less dramatic effect. You know, if a hydrophobic amino acid gets replaced by another hydrophobic amino acid, it might not change the folding of the amino acid, so it might not have as much of an impact if a hydrophilic replaced a hydrophobic. Okay, and the last one is a nonsense mutation, and this is where the change in the DNA sequence causes a premature stop codon. So it stops the protein really early, and this would cause a pretty dramatic effect because you're just missing a huge chunk of the protein. All right, so when it asks for types of proteins or types of mutations, these are the things that it's looking for in your packet. All right, 2 examples it mentions, and we may talk about these in more detail in the future, but just enough to answer your questions for now, is sickle cell anemia and cystic fibrosis. These are both mutations that cause a genetic disease. Okay, sickle cell anemia is just a point mutation, one base different in all our bases, but it causes the hemoglobin protein to fold differently. So it doesn't carry oxygen in the body as well. And it also, that change in the protein causes a change in the red blood cell shape, hence the name sickle cell. You can see those like sickle-shaped blood cells in that picture in the center there. Okay, sickle cell is an autosomal recessive allele. So if you got one from mom, one from dad, you would end up with sickle cell anemia. And your prognosis is a pretty short life because those sickle cells can cause blood clots, stroke, and heart attack. And also you're just less able to bring oxygen to your body, so that can cause your body to shut down as well. Now, people that have sickle cell can get blood transfusions every 3 or 4 months, and that's what they would do in a more advanced country as far as medically advanced. I actually went to college with somebody like that. So you are dealing with a long-term commitment there. There has been some interesting new research in sickle cell right now, but that's what I'm going to leave it at right now. The interesting thing with all genetic diseases, you have to think, why is it still in the population? Because if it was just detrimental, typically through natural selection, you're going to see that decrease in the population, especially if it's something that occurs before reproductive age. What's interesting with sickle cell is it's what's called a heterozygote advantage, because if you have one normal allele for normal blood and one sickle cell allele, it changes your blood just enough so that, you can still bring oxygen to your body pretty well. You're not going to have as much sickling of the blood. You're going to function pretty normally. However, it changes your blood enough that you become immune to malaria, carried by those nasty little mosquitoes on the right. And so if you are immune to malaria, that's a huge advantage in certain areas where malaria is carried. It's more of a tropical disease. We don't see it here. The mosquitoes wouldn't survive here. So that's why you see sickle cell high in populations from sub-Saharan Africa, especially. Okay, so there's a little on sickle cell. Cystic fibrosis is a mutation as well. It is a deletion mutation. It actually is a deletion without a frame shift. It deletes 3 bases, the most common cause. And that causes a transport protein in the cell not to function properly. It's one of the transport proteins that helps chloride ions move out of the cell. Okay, and because this, and water as well, but because of that decrease in movement, you have a whole series of things that occur in the cells. And basically that is, you start to get this like thick mucus buildup on the outside of the cell linings, especially in your lungs, but it can be other places in your digestive tract, pancreas as well. And then that mucus can harbor bacteria and cause secondary infection that can be very dangerous. When someone is born with this, you have this, the common thing that people do is they kind of hold their child with their head down, cup their hand, and kind of hit their back multiple times a day to try and flush out some of that mucus to reduce the chance of infection. But there's a lot of complications that can occur from this disease. It is also an autosomal recessive disease, so both parents had to carry it. And we're not really sure. It's fairly rare, but it's pretty common in certain Northern European populations. It's more common in. And in some of the populations where we saw high amounts of tuberculosis. So one of the thoughts is that maybe this was an advantage to reduce tuberculosis back in the day when that was a huge killer. Anyway, there's your little bit of info that help you with those questions. Work on those throughout the beginning of this week. So Monday, Tuesday, and Wednesday, I'm going to be listening to simulations, and you guys are going to be finishing that up and finishing up the questions. Okay? Have a great day. Talk to you later.